And I think you might be muted, Michael.
Is that better? Can you hear me?
Yep, we can hear you now.
Okay. Hello, everyone. My name is Michael Moore. I'm the manager of the Oscar Imaging Facility located in Dover, Delaware. We have
Oh, somehow you muted again.
I see that the space bar actually mutes me. Okay, sorry
Okay,
about
there you
that.
go. You're fine.
Okay. We're located at Delaware State University. The director of the facility is Hassan Bukhari, and I'm the manager, Michael Moore. Um, So we're located on the very first floor of the building. So if you've been to the Oscar building, there's a total of 3 floors. The first floor is dedicated to the imaging facility and one research lab. Um, and then on the second and third floors, there's just various optics research from material science with light detection, but also with advanced microscopy techniques throughout those labs. Um, it's a nice little imaging suite. We have individual rooms, so you can slide the door. There are sliding doors. You can shut the lights off if you need privacy, or to keep the lights down low for the imaging. And it's a pretty tight little um, facility that has a lot of resources in it. So right now, what we have in our light microscopy lineup is a brand new is the Pressed Optics spinning disc confocal. It's the X-Lite V3 spinning disc confocal. We have Wrap Optics FRAP system on this. We got this July 2022. It was through a COBRE subaward for instruments. I think we've applied for that before in the past, and we've never gotten it. This was the first time that we've gotten this. It's on a IX83 Olympus microscope, and we have some pretty high-end objectives, and it's ran through the software called VisiView. And most of the imaging that's done on this is live cell imaging. So if you know anything about spinning disk, it's a high-speed imaging modality. We can do dual color, green and red, or far red and like a yellow color. We can do GFP, YFP. We can do RFP. Pretty much anything that you can think of. Fluorophore-wise or fluorescent protein-wise, we can pretty much do on this system. Plus, we can do FRAP experiments. A lot of the experiments that are done on here is calcium imaging. The next instrument we have in confocal is the Zeiss point scanning confocal. This is a LSM 780 confocal. This was donated to our facility, and it's on loan from University of Delaware around May of 2022. So some of you may be familiar with this system. The 980, I can't remember what building it's in, the CCI, I can't remember. In that building, they have the 980, which replaced this. The nice thing about this system is it's got full spectral capability, so we can spectrally unmix the samples on a pixel by pixel basis. Plus, we can do fluorescence correlation spectroscopy. My hope is to eventually get a 2-photon laser for this system so that we can start doing 2-photon microscopy on it. I have all of the optics plus detection. I just need a laser. So hopefully that might be a grant coming down the pike that we can apply for to get. This was some images that were taken on the LSM 510 confocal back, this has been a couple of years ago. We had an LSM 510 on loan from University of Delaware, and this was detection and localization of protein interactions between wild-type CD44 intracytoplasmic domain and the transcription factor RUNX. And what we did was we did the Z-stacks on the system, and then I took these Z-stacks, did some volume rendering on them, and then bot analysis through the IMERA software. And we were able to detect, by creating masks, what was in the nucleus or on the surface, and then we could determine what was outside, and then they were looking for ratios of these markers from inside and outside the nucleus. So it was a pretty nice little project for a graduate student, and just one of the capabilities of things that we have done in the facility. So we have a scanning electron microscope in our facility as well. With this system, we mostly do material science, but we also can do some biological work. I don't have the full suite of sample prep materials and sample prep equipment to do full biological SEM work, but we do have a gold coater, carbon coater, so that we can make things conductive. There's a few smaller pieces of equipment that I need to do full biological samples in a classic high vacuum SEM environment. But what we do have is we have environmental SEM. And if you're not familiar with that technique, it's a very powerful technique. And I believe we're the only facility in the state of Delaware that has an environmental SEM. I don't think there's one available at UD. They could get the capability, but for some reason they just haven't found a lab that has one. In environmental SEM, we can take the sample that's in its biological state without any sample preparation and put that into the SEM. And under a compromised type or lower vacuum conditions, we cool the sample down, and then we can collect the image through backscatter through a beam skirt. And when we do this, we can get very high contrast, very high resolution images of untreated samples. This works wonderful with samples that are botanical, seeds, plants, mostly plants, insects, anything you can pull out of the environment. I've even taken pond samples and have cooled them down, and I've taken images of these types of samples. Cells, a little harder to work with. Things like yeast and bacteria is possible, but it's very difficult techniques involved with doing that. So on the left here, the RE lab from the agriculture department at DSU had a visiting PhD student, and she was looking at yam starch granule size over periods of time through different storage techniques. And this is an environmental SEM image of starch granules. It's extremely high resolution, and we were able to do a particle sizing with a program that we have called Topo Maps 3D. And then on the right here, we have these iron oxiding Zetaprotobacteria, and these were taken from a local stream in Dover, Delaware. If any of you are familiar with Clara Chan at UD, she's done, I don't know if she still does, but she's done a lot of work with these types of bacteria that create these iron oxide tubes. And when I used to work at UD, we did a lot of imaging, and for them to do SEM imaging of those tubes, it went through a lot of sample preparation, and it was very difficult. This actually, I just took a few drops out of a stream into a test tube. I brought it back, dropped it onto the Peltier stage, cooled it down. I was able to get the same images without any sample preparation. So it's a very powerful technique. And these are just diatoms that were collected from the Blackbird Creek part of the water. Is a water quality study that the Osbay lab was doing from the ag department. So most of my SEM imaging comes from the ag department or the material science areas in the physics department. We also have atomic force microscopy. So we have a Bruker Innova AFM. That technique, if you're not familiar with AFM, it's a touch technique. It's a technique where a cantilever comes down with a very sharp, what we call a stylus, kind of like a record player, comes down to the surface of a sample. The sample is then moved back and forth in a lateral direction, and then you can collect the topography information. Plus, you can get atomic force information, pulling, pushing, indentation, how hard a sample is. A lot of the techniques that are done on the atomic force microscopy is heavily material science, but that doesn't mean you have to do just material science. There are biological, we have a liquid cell for biological samples. So if you want to look at stuff like collagen fibers, you want to look at bacteria, you want to look at flagella, you want to look at, you want to take any kind of data from protein interactions, you can do that on an AFM. It's difficult, but it can be done, and some of the information is very powerful. So, it can open up a lot of passageways into other areas based on force interactions between biological molecules. People look at protein interactions at the membrane surface. So you can take a stylus and usually they'll use things like styrene beads that get treated with another protein, and then you can bring this into contact with a cell, and then you can look at the forces of attraction or if there's any kind of binding. It's a very difficult technique to do, but it is possible on this system. We also have a Bruker Nano IR FM. This is a top-notch instrument. It has the ability to do AFM mapping of a surface and simultaneously do infrared spectroscopy at point by point on the sample in a nano spectroscopy way. So you can get nano IR information from the sample on any scale that you're scanning. So we can scan down to a scan area of about 1 micron. So you can imagine your field of view being about 1 to 2 microns. You can get IR information from discrete nanoscopic regions on your sample. We can do tapping and contact mode just like any other AFM. There's a technique called SNOME. It's kind of like it's a near field microscopy technique. I'm not 100% familiar with the technique myself. I'm still learning in this area, but what this can give you is contextual information about the phase differences between a sample. So it's using an IR laser along with this microscopy technique in order to give you these phase shifts, phase differences, or if you have ordered or unordered amorphous crystalline structures. So it's again, that's more on the the material science side of things. We also have a spectroscopy core that's connected to us, and we do FTIR. We have a Bruker Alpha 2 FTIR. We have a Hariba macro RAM. That's for bulk spectroscopy of a sample doing Raman. If you're not familiar with Raman, it's a technique that's been around for a very long time, but it's getting a lot of publications lately due to the fact that we're understanding it better. But detection techniques have become much more sensitive, so we're able to detect the Raman spectra a lot better than we used to in the past, and that's becoming a very relevant technique because we can create very discrete signatures for different types of molecules, proteins, carbohydrates, DNA, almost anything you can think of. We can extract a very distinct Raman signature for that sample. In a bulk spectrometer like this, we're looking at samples that are either in a cuvette or on some type of substrate. So this would be really difficult to do for biological samples, but nonetheless, it's a very powerful technique. So we do have plans for future equipment. The equipment that will be arriving soon is we are getting a Hariba Explorer Plus Raman confocal, and that's slotted to be delivered June 2023. So this concept is very interesting because you're taking a microscope now, and you're able to pinpoint a particular region or area on your sample. You can then focus in on that area, and you can do Raman confocal spectroscopy on that particular area at very high magnification. What this means is you could look at biological samples now, and you can get Raman confocal images. You can get or fluorescent images, and at the same time get your Raman spectra. This is where confocal is probably going to migrate to in the next 10 years, where we will be using more Raman scattering as a way to detect samples that aren't labeled. You do not need labeling for your sample when you're using these techniques. So if you have a protein library for Raman spectra, you could literally do a confocal scan of your sample, collect that and label all those proteins that came up within that Raman map. If you wanted to look at protein-protein interactions, you could then start parsing out the different spectra that come from that Raman confocal map and seeing what is correlated together and what is not. So this is becoming a very important technique in the pharmaceutical realm. They use it a lot for looking at the purity of different pharmaceuticals, but also it's becoming very, very popular in biological samples, not just materials. We will be getting a light-directed infrared spectroscopy, and it's called an LDIR, and installed sometime in August from Agilent. This will be mostly used for microplastics. So microplastics are becoming a very important and very hot topic in environmental science, also in biological sciences. And there's several groups on campus that are looking into microplastic research. The Raman confocal also will do microplastic and particle finding. So it'll be able to you can look into cells and be able to see if there's microplastic signatures within cells. The Agilent UV Vis IR carry, the 6000i, we are getting that installed in August as well. So it's basically going to be a high-end UV Vis with near IR capabilities. Also arriving in August is going to be our Agilent Citation 10, and we will be getting that with a BioSpa. So the specifications are here on the right-hand side. It has automation capability through the BioSpa, and the camera that's used on it is going to be a Hamamatsu, the Orca line. It's a little bit older camera because they've definitely moved up over the years, but it's still a very, very good camera, this SCMOS. It'll have the 4-zone incubation. You can move it with a joystick. You can do montages. You can do full confocal fluorescence. You can do bright field. You can do wide field, phase contrast. Pretty much any light microscopy modality that you can think of, you can do on this system. It's designed for high throughput, so if you have multiple samples and you want to look at multiple trials, multiple different treatments all at the same time, with the BioSpa, you can put those in glass bottom dishes. You can do all your treatments. You can have them preloaded, pre-programmed. It will put it in the BioSpa. It will warm up your sample, keep it at 37 degrees, and CO2 mix. And I've been told by Agilent that there have been some groups have done week-long experiments in these conditions with media changes, treatment changes, removal of drugs. So this is going to be a very powerful instrument to have on campus for anyone who wants to do long-term imaging studies of biological processes. And we are getting the system fully loaded, so it will have, I think, almost everything that you can get on the system. The last piece of equipment that should arrive maybe in August or September is we're getting the NovaCyte Penteon flow cytometer. This is a 5-laser system. This is an area outside my wheelhouse, so I'm going to have to do a lot of learning on flow cytometry. So I don't know a lot about it, but I've been told that this is a very, very nice system. And once it's installed, I get training and I'll know a lot more about it. Then I probably can talk a lot more about it, but I know that it's going to have a lot of capabilities. What's being installed right now, actually starting today and then throughout the end of May, is we are getting a deep sim super resolution microscope from Crest Optics. So this is a very large field of view. It has a lateral resolution of about 100 nanometers. And the axial resolution about 300 nanometers. So if you're not familiar with super resolution, structured illumination, or SIM, is one of the easier ways to do super resolution microscopy. It doesn't require any special sample preparations. It doesn't require any special fluorophores. It doesn't require any special transfections or special antibodies or fragmented antibodies. A lot of the other techniques that you might hear in super resolution and see very beautiful and very compelling images with lots of data, a lot of those techniques are actually very difficult to do because of the sample prep or because of how you are treating the sample ahead of time in order to get those super resolution images. Structured illumination has been around for actually a very long time. It's one of the very first super resolution techniques, but when all these other techniques came out, such as STORM, D-STORM, what's the other, Palm. These are stochastic techniques where you're looking at lights blinking on and off, and then an algorithm later on takes and reconstructs the image. And these, in many ways, are breaking the resolution limit, but they're still bound by that optical system that is diffraction limited. And so this is still a diffraction limited system, except you're just modulating the light with a grid pattern or with a micro lens array. In the case of this is a newer technology using micro lenses. And these micro lenses are able to create like a micro lens mask. So you can think of, I should have had a picture of this on here, but I didn't. You can think of it kind of like a square with a bunch of little dots in it, and all those dots are illuminated. And then you have another mask on the emission side. So that's the excitation side. And on the emission side, you have another group of dots that align up with that for whatever color you're working with. So if it's green or red, those have to be aligned with the excitation emission source. And then these dots are then modulated in a particular pattern. I don't know how they're modulated in the system because it's proprietary. It's part of their patent. But however it's modulated, they keep track of the modulation and the rotations on these micro lenses and masks. And then later on, you take that dot information and it's then reconstructed through an algorithm. And this isn't a real-time super resolution. It does need post-processing. But because of the acceleration in the GPU market, we now have graphics cards that can almost give you an image within just a minute or less, even very large complex Z stacks. This system is really nice because we at Dell State will be the first I've been told by the company in the world to have a standalone super resolution like this. So the picture you're seeing here is how it will look in our system. Usually this is piggybacked, this Deep Sim, which just came out last year, is piggybacked to the spinning disk system. So I kept requesting, why can't you do this as a standalone? And they kind of let me know, well, we've been working on that. We have to get through some FDA regulations. And there were other universities. I know that Pittsburgh at the Center for Biological Imaging has multiple Crest Optics confocals. They were looking at getting a standalone, so they had been requesting. I think at Princeton, there are a couple other places. And it just so happened RPO got in before anyone else. So we're the first install of it, which is very nice. And so this system will be used to do, we can do live cell super resolution imaging on it. We'll be able to do just fixed slides. We can do live cells on it. We can track organelles in super resolution. So it's going to be a very versatile system for it's going to cover a lot of projects here at Dell State, but hopefully other projects across the state of Delaware and other institutions. Last but not least, we do have a wet lab. We have the ability to do tissue culture right here next to the imaging facility. So if you do want to come to the facility and you want to bring cells, you can bring them ahead of time, get them acclimated. So if you're traveling a long distance or you're coming from across campus or somewhere else, you can throw it in the incubator. And we also have a lot of different sample prep equipment for material sciences, like the gold and carbon coater. We have a lot of image analysis software packages. So we've got Imaris and Aviso, Huygens deconvolution, Velocity, TopoMap. Some of these you may have heard of. So we have full licenses for all of these different packages. We also offer the free packages, the ImageJ and ZenBlack. We just got the license for ZenDesk, so I can look at images that were produced on confocal on a Zeiss system in its raw format and manipulate it there. So, and that's pretty much it. Any questions?